Hello, Pima Aztecs. Welcome to Chapter 16, Adaptive Immunity, Part 1. So I think there's 4 videos for adaptive immunity. So this first video is just an overview. Here's our flow chart. So we've gone over the innate defenses. That was Chapter 15. It's also called the non-specific immune system. And so for Chapter 16 is the adaptive defenses or the specific immune system. Well, let's come over here and look at the check marks. It says adaptive or specific immunity is also termed acquired immunity. Boy, how many names can they give one thing? So down here, it's adaptive immunity, it's specific immunity, it's acquired immunity. They're all the same thing. But you get adaptive immunity or acquired immunity when you are exposed to antigens. And we'll talk about what those are. And then the white blood cells, called the B lymphocytes and the T lymphocytes, are the primary cells involved in the adaptive immune response. But you can get some cells coming over from the innate defenses, like the macrophages, natural killer cells, dendritic cells can also play a role in the adaptive or specific immune system. Adaptive immunity is also called the third line of defense, and there's 5 distinctive attributes of the third line of defense, and we'll talk about those 5. But let's look at this cartoon first. So here's a macrophage, this guy, and here's his pseudopodia coming out from the macrophage. And right here, this little blue circle, that would be a Toll-like receptor recognizing either a PAMP, pathogen-associated molecular pattern, on a bacteria. So here's the invading bacteria. Or it could be recognizing something more specific. So, but in this cartoon, the macrophage recognizes the invading bacteria with its Toll-like receptor, seeing the PAMPs on the bacteria. Then the macrophage ingests the bacteria. So here's the bacteria inside. And remember, there's a lysosome and a phagosome, a phagolysosome. They merge and then they digest the bacteria. And interestingly, what happens is pieces of the bacteria are cut up and then they are put here on the outside of the macrophage, and now the B cell or B lymphocyte with its B cell receptor, so this black thing is B cell receptor, can recognize these specific parts of the bacteria. So this is a little tricky. The macrophage recognizes the bacteria non-specifically via the Toll-like receptor PAMP interaction, and then the B cell specifically recognizes certain pieces of the bacteria that the macrophage presents on its cell. And then once the B cell recognizes those specific pieces of the bacteria, it gets activated, becomes a plasma cell. Plasma cell secretes antibodies, and now those antibodies will go out into the blood, and the antibodies will recognize those specific pieces on the bacteria that were presented to the B cell receptor. So really the B cell receptor, this thing, is almost identical to the antibody that the B cell makes. Okay, so now we'll go back to 5 distinctive attributes of the third line of defense. The first attribute is specificity. So in the third line of defense, the immune response is only against one specific molecular shape, and that's what this cartoon shows. So this one specific molecular shape that was a piece of the bacteria was presented out here on the outside of the macrophage, and it was specifically recognized by this B cell receptor. This B cell receptor will not recognize any other molecular shape except the one that it recognizes. That's specificity of the third line of defense. The second attribute of the third line of defense is inducibility. So cells of adaptive immunity are activated or induced only in response to specific pathogens. So for example, this B cell could, with its B cell receptor, could try to recognize, let's say, this molecule, this square molecule. If this B cell receptor, this black thing, the B cell receptor doesn't recognize this thing, then the B cell will not become induced or activated. The third attribute of the third line of defense is clonality. So once induced, so for example, once this B cell got induced, because it bound to this specific shape, there's a specific antigen, then this B cell, it doesn't show in the cartoon, but this B cell will form a clone of cells, a clone of plasma cells. So it just shows one plasma cell in this cartoon, but it would form, the B cell, once activated, would form a clone, so hundreds of thousands or millions of these plasma cells, making billions of antibodies. The third attribute of the third line of defense is unresponsiveness to self. So if this were a B cell in your body with its B cell receptor, it should not recognize anything that is a self molecule or a self antigen on any of your cells. This B cell receptor should not recognize anything that's on any of your cells. The fifth attribute of the third line of defense is memory. So the third line of defense remembers things. So in this cartoon, it shows that once the B cell is activated, it forms a plasma cell, actually a clone of plasma cells. But it doesn't show in this cartoon, but it also forms a whole bunch of memory cells. And then if this bacteria with its antigen comes into your body again, the memory cells will be activated, form antibodies, and kill the bacteria before you even know you're sick. So there's two types of lymphocytes. A lymphocyte is a type of white blood cell. There's T lymphocytes and B lymphocytes. We call them T cells and B cells for short. So this is a cartoon of a B cell. Here's a cartoon of a T cell. And here's the protein receptor. So this is a B cell receptor. We just saw that in the last cartoon. This is a T cell receptor. These are made of proteins. That's why they call them protein receptors. And they're stuck in the plasma membranes. And these receptors, B cell receptor and T cell receptor, recognize specific pathogens because they have foreign or non-self molecular shapes called antigens. So in the previous cartoon, we saw this B cell receptor. The B cell receptor recognized only a specific shape or antigen on a bacteria. You can also, B cells and T cells can also recognize toxins this way. And this is a light micrograph of a lymphocyte. So this could be a B or a T lymphocyte, I can't really tell. And then here's red blood cells. B and T lymphocytes form from blood stem cells in the red bone marrow. That's RBM, red bone marrow. So they form there, but then for B cells to become mature, they stay in the red bone marrow. And if you are a T cell and want to become mature, be able to go out and fight all the bad bacteria in the world, you have to leave the red bone marrow and you go to the thymus to mature. So this is a cartoon talking about antigens and epitopes. So this gets a little tricky because if you're not an immunologist, I think you kind of use antigen and epitope interchangeably, but they really are different things. But your adaptive immune response, or the third line of defense, the cells are not working against a whole pathogen. The B cells and T cells of the third line of defense work against a portion of the pathogenic cell. That's the antigen and the epitope. And the B and T cells also want to recognize these antigens and epitopes as foreign, aka non-self. Because then if they start recognizing your self antigens as non-self antigens, then they mount an autoimmune response. So here's kind of the difference between antigen and epitope, but you can use the terms interchangeably. So here's this whole guy is a bacterial cell. And see these little shady parts? These are actually the antigen parts. And then these little specific shape, those are the epitopes. And those epitopes are also called antigenic determinants. So lots of nomenclature in there. And then here's this cartoon, it just shows this specific antibody that was secreted from a plasma cell. See how it's the shape of it here? It recognizes this triangle shape. So that's antigens with epitopes. And sometimes antigens and epitopes will be used interchangeably, and sometimes they won't. Adaptive immunity consists of two basic characteristic responses. So if you're attacked by bacteria, what are the two basic types of responses your immune system can mount? You can do, first one, humoral immunity. So humor just comes from the body fluids, which in Greek, those are the humors, your body fluids. And in humoral immunity, that's when the B lymphocytes make antibodies, and then the antibodies are floating around in the body fluids like your blood, aka your humors, and the antibodies act against extracellular pathogenic antigens. So what that means is if you are a bacteria and you have pathogenic antigens on you, and you are not inside a cell, but you are roaming freely around in the tissues of the blood, then B lymphocytes will produce antibodies that can kill you because you're not inside a cell. So in this cartoon it says, here's our B cell with its B cell receptor, and the B cell is saying, I attack invaders outside of cells. So if you are an invader and you get inside a cell, well, don't worry, Mother Nature devised another arm of adaptive immunity called cell-mediated immunity. So, and in this cartoon it says that this T cell attacks invaders inside the cells. So let's say one of your cells in your body was infected with a pathogenic antigen on a bacteria. Now that bacteria with its pathogenic antigen is inside a cell. T cells can find that, which is pretty cool. And we'll talk about that in detail when we talk about how T cells find foreign invaders that are hiding inside your cells. Okay, this cartoon is concerned with auto or self antigens. So in this cartoon, so here's some normal body tissues. Here's normal cells. They've got the nucleus here, and then they've got these antigens or self antigens on the outside. Here's a bacteria. It got attacked by antibodies. Yay, that's what's supposed to happen. But in an autoimmune disease or disorder, the antibodies these specific antibodies will attack these specific epitopes on this bacteria. But these antibodies will also, look what happens, they also attack your cells. That's an autoimmune disorder. So your body has to closely regulate adaptive immunity, or you can get an autoimmune disorder. Okay, the lymphatic system. Let's talk about this a little bit, because B cells and T cells travel in the lymphatic system, and in the lymph nodes, that's where B cells and T cells really do their work. So probably had this in 202. You've got all these lymph nodes. So for example, when you're sick, they'll touch your cervical lymph nodes. Also here in your inguinal lymph nodes, you have axillary lymph nodes. And then what they do is they take the lymph capillary here in green. This is a lymph capillary is next to a blood capillary. And then all this stuff out here, this is extracellular or tissue fluid. And if a bacterial invader gets into the lymph capillary, that lymph capillary can take the bacterial invader to all these different lymph nodes. So we see got a lot of nodes strategically placed. And in those lymph nodes, guess what's there? B cells and T cells ready to recognize the specific invader. So the lymphatic system, that's where you produce, mature, and house adaptive immunity cells. Those are B and T lymphocytes. In the lymph nodes, you'll do antigen recognition. So the B and T cells will recognize the antigen in a lymph node. B cells will make their antibodies. And then there's also chemical signals and mediators that go along with this whole process that we'll look at later. So the lymphatic system basically is a surveillance system. It screens all the tissues of the body. So you've got capillaries here in all tissues of your body, and you've got lymph capillaries in all tissues of your body. But you have special lymph tissue at entry portals in your body, like your throat and your intestinal tract, so that, well, that's the most likely place for bacteria to come into. So you've got to have extra heavy-duty surveillance system there. Okay, primary lymphoid organs are here: the thymus gland, the spleen, red bone marrow. But once lymphocytes arise in the red bone marrow, then B cells stay in the red bone marrow and mature. T cells go to the thymus and they mature there. T cells mature in the thymus. And then from there, from the bone marrow and the thymus, they go to these secondary lymphoid organs like the lymph nodes. And then they stay there. Then you just have a bunch of B cells and T cells in your lymph nodes, ready to do battle. And this is a cartoon of a lymph node. So this outer region here is the cortex, and then the inner region is the medulla. You probably remember this from the kidney. And then you have, this is a lymphatic nodule. And within the lymphatic nodule, you have a germinal center where B cells and T cells can proliferate. There's thousands of lymph nodes in your body to facilitate interaction among immune cells. So like I said, the lymph nodes house the lymphocytes in a lymphatic nodule, specifically the germinal center. And the lymphocytes are looking for foreign molecules. So the lymph will percolate. So the lymph will come in here, and it will percolate through the cortex and the medulla. And then as the lymph flows past these B cells and T cells in the germinal center, the B cells and T cells with their receptors can kind of interrogate that lymph fluid for bacteria. The lymph node is where the clones of B and T cells replicate. So when you get sick and your lymph nodes are large, that's because you've formed a clone of B cells or T cells. Other lymphoid organs we talked about in the portals: mucosa-associated lymphoid tissue, or MALT. There's respiratory-associated lymphatic tissue or lymphoid tissue, that's RALT. There's gut-associated lymphatic tissue, or GALT. So there's lymphatic tissue is strategically placed all over your body to fight off the foreign invader. Okay, here we've seen this cartoon before. Here's our bacterial cell with the antigen, and then the little shapes are epitopes. That's a bacteria cell. But here's one of our cells. So this would be a cell in our body. Here's plasma membrane. Here's the nucleus. And then sticking out of the plasma membrane, we have antigens too, but they are called self-antigens, or autoantigens. And if you are a normal uninfected cell, you've got normal autoantigens sticking out of your plasma membrane. If, however, you get infected, you will present the non-self antigens in your plasma membrane, and then your adaptive immune system can take care of it. We'll talk about that more in detail later. Okay, I like this. This is a cartoon of an antigen. Antigens are usually proteins. So this pink thing, this is the antigen. And then look, it's got these yellow parts on it. Those are the epitopes. This is kind of a cool, instructive cartoon. Epitope 1, 2, 3, and 4 on an antigen. So the body or your cells recognize antigens by their three-dimensional shape of these regions called epitopes on an antigen. And the epitopes are also called antigenic determinants. If you are a really large molecule, like up to 100,000 Daltons, you're a better antigen than a smaller molecule. Proteins and glycoproteins are good antigens. So then here's our cartoon again, the antigen with the epitopes on it. And then look what happens. Each of these antibodies, antibody 1, antibody 2, antibody 3, and antibody 4, those antibodies only recognize those specific epitopes on the antigen. If you are a complex molecule, you make a better antigen than a simple molecule because complex molecules have more epitopes. And remember, the epitope, that's a part of an antigen that is recognized by the immune system cells. So this is just another cartoon. We saw this one before. Here's one of our cells. Here's the nucleus. They cut out this square, and then they show you, here's the phospholipid bilayer, or the cytoplasmic or plasma membrane. And then you see this antigen is stuck in the cytoplasmic membrane. And then here are these specific shapes. They're calling those are the epitopes or antigenic determinants. Good antigens can include the components of a bacterial cell wall. So let's go down here and look at this. Here's a bacterial cell wall. Here's its plasma membrane. Here's its peptidoglycan, so its cell wall. And then here's another bacteria. Here's phospholipid bilayer. Superficial to that is a smaller peptidoglycan. And then you have this outer membrane. You guys remember which one is gram-positive and gram-negative? This guy over here is gram-positive. And this guy over here is gram-negative, because gram-negative cells have an outer membrane, gram-positive cells don't. Okay, now let's make antigens even more complicated, shall we? Let's have exogenous antigens and endogenous antigens. So in this cartoon, here's an extracellular microbe, and it's got here's an extracellular microbe too. This kind of looks like a virus, and this guy looks like a bacteria. But anyway, they're extracellular microbes, and sticking out of their cell wall or their plasma membrane are antigens, but they are specifically called exogenous antigens. Oh, and then also this bacteria looks like it has I don't know, this could be a flagella probably, and then it's got these specific epitopes on it, epitope antigen. So these are all exogenous antigens. So exogenous antigens include toxins and other secretions of microbial cells. Exogenous antigens include infectious antigens, so they would include the components of microbial cells. Like the cell wall could be an exogenous antigen, the membrane. This flagella with its epitopes would be an exogenous antigen, and the conjugation pilate could also be an exogenous antigen. And this is kind of tricky. It says here extracellular microbes are exogenous antigens. More specifically, extracellular microbes have exogenous antigens in their cell wall and plasma membrane. Those are infectious antigens. If you are a non-infectious antigen, you would be like pollen, cat dander, peanuts. So those would not be infecting antigens. So in contrast to exogenous antigens, you have endogenous antigens. So here's one of your cells in your body. There's the plasma membrane, there's your nucleus. This is a virally infected cell. So the cell, the virus got into your cell. Now it's an intracellular virus with its epitopes and antigens. Now this intracellular virus would be classified as an endogenous antigen because it's inside your cell. Endogenous antigens, like I said, are inside body cells. You could be protozoa, fungus, bacteria, viruses. Once they're inside your cells, they can reproduce and make more themselves or more endogenous antigens. If you are an infected cell, that means you have endogenous antigens in you. Okay. And then, so in addition to exogenous antigens, endogenous antigens, there are autoantigens, and we already talked about that. So autoantigens are just normal self-antigens. You should not mount an immune response against your autoantigens. If you do, that's an autoimmune